I'm glad to introduce our third speaker, Dr. Tina Yu, from the Regulatory Affairs Department of ProBio. Tina has more than 17 years RA experience in biotech and pharma industry. She is experienced in regulatory affairs, GMP inspection, and project management. She led to obtain over 100 R&Ds and over 10 BLAs from FDA, EMEA, NMPA, etc., including MAP, bispecific, fusion protein, ADC, CGT, and etc. She participated in multiple PLI on-site inspection by FDA, EMEA, and NMPA as well. In this talk, Tina will discuss the topic about CMC regulatory considerations for CGT products. Now, I will hand it over to Tina.
Hello, hi, can you hear me?
Yeah, we can hear you.
Okay, so let me share my screen.
Okay.
Can you see my screen?
Yeah, it's good. You can start now.
Okay, thank you. Thank you. Good afternoon, everyone. Thank you for attending today's meeting. I think especially thank you for some audience already listened for several hours. So I will just have a very quick talk on some CMC regulatory consideration on CGT products. Sorry, let me see. Okay. Yeah, this is my today's topic, some CMC consideration for IND stage, and also some work scope, CMC work scope for BLA application. And the last part, I would like to talk something about compatibility consideration. Okay, let's. This is very common and overall picture of the regulatory activities during the product development. From this figure, we can see that during the product development, we have the IND application and the BLA application to get the clinical trials ticket, and also to have the approvals for your product commercial. So during the, no matter for the IND application or BLA application, we have a lot of activities to be done, including the CMC or non-clinical and clinical. So this table also shows us that during this period, for from regulatory perspective, we have some activities like after IND application during the clinical trials, we can think about whether this product or whether this indication can get orphan drug designation or breakthrough therapy designation. And before the BLA application, we as a RA, we will think of whether we can get the priority review or fast track or other pathway. All of them can speed the filing procedures and can make sure our product can be launched as soon as possible. And also, it shows that during for each stage of works, that we have to follow some requirements. Like for the preclinical studies, for the toxicology study, we need to follow the GLP requirement. For the manufacturing, the GMP requirement need to be complied. And also for GCP, that is for the clinical studies. So a lot of requirements during the production or studies, and also every stage work costs a lot of time. So how to develop the strategies for each stage of work, and also the filing strategy is very important for product owner. Okay, so as a manufacturer of the product, our goal is to provide an affordable, safe, and effective drug with consistent quality. So for each stage of work, we have to we have our specific goals. Like for the phase 1 studies, safety is the most important. But when the product go to the phase 2 or phase 3, except the safety, we need to more consider on efficacy. So yeah, so today I will focus on to talk more about during the IND stages, the safety is what we need to be considered. So CMC's review principle for IND is safe and consistent quality. To make sure we have the safety product, these items we need to pay attention. The first is the control of materials, especially if the material is the biologic source. And then the manufacturing process. The manufacturing process should be well designed, and the in-process control is in place during the manufacturing. And also the testing and the characterization on product and the intermediates should be there, and the testing method accepted criteria should be set up. Let's go over one by one. For compared to small molecule, for biologics, there are more biological source material used, such as host cell expression vector, stable cell line, and the cell banks, and all other raw materials. So when we submit to the IND, we should make sure that there are sufficient information in the IND package. Like for the host cell, we need to make a very clear history explanation on the package to show that to agency that there's control the risk. The risk is very low. So we need to provide the testing and the characterization result, which is comply with the requirement. And for the expression vector, how you design that one, and the sequence is confirmed. So this information should also be shown to them. And for the cell line, how develop the cell line, how you do the screen is whether it's the what's the clonality data about that one, and should also be shared. So also because as we mentioned that we want to produce the consistent quality product, so we don't. It's not expected to see that there are some mutation after several passages of cell run. So we needed to do genetic stability studies for the cell line stability. We recommend to do these studies at the early stage, because if we don't have that data, even we get the IND approval, but I think the agency may ask, but even we get during the clinical stage, if we find that there are some mutation or the sequence is changed or some issues, then we need to consider the changes the cell line. That will be a big change. We need to do a lot of compatibility studies to demonstrate that the cell line change will not impact the product safety and efficacy. Or else we need to maybe trigger additional studies. And for the cell banks, yes, because we want to continuously produce the product, so cell bank always be established for before GMP production. So also for cell banks, we need to do a lot of testing and characterization based on like ICH guidance or each region's specific requirement. And also for cell banks, because it will be used for many years, so we will store the cell banks at appropriate temperature. So the storage stability needed to be conducted. And yeah, except these materials, we have other raw materials used during the process. We need to analysis whether they are animal or human origin. If so, we need to analysis whether there is any TSE, BSE risk. Except these biologics origin materials, we have a lot of other materials. These items need to be considered. That the first one is we, as the product, we want to have a high quality, right? So the materials also need the highest quality to meet the requirements of from technical perspective, such as we want to have the qualified the media or growth factor and so on. For these materials used for the GMP manufacturing, they should be purchased from the qualified vendor. So before that, we have to establish a vendor qualification program. Except that one, yeah, for each materials, the vendor has vendors have some their own specification, but as a manufacturer, we also need to set up appropriate incoming material qualification to release these materials for production before after we purchased these materials. And another point is that we need to make sure there's sufficient supplies of critical materials. As you know, several years ago, there's a COVID-19 pandemic. And also recently, we talk about like the tax rate increase a lot. All these things may impact the materials supplement. So as a manufacturer, we need to make sure there's sufficient supplies choice, so we can, if there's any problem, we can change the materials. And also, as we mentioned, that our goal, one of the goal is to produce affordable product. So cost is also our consideration for the product development. And yeah, after the materials, there's some, we talk about the manufacturing process design and the control. I think we already talk a lot. My colleagues talk about the manufacturing, about the plasmid, and also LVB. I show this picture just as an example. For plasmid and for LBV, we all have the upstream production, downstream production, and if there's DP product, no, even the plasmid or LBV, it can be as a DP. So except the upstream, downstream, we have the fill and finish. So during the production, we control the environment, we have appropriate equipment, we have qualified materials, and also during this, we also have the in-process control or in-process monitoring for some steps to make sure the final DS or DP has will be qualified or meet the acceptance criteria. Regarding the manufacturing, not only just as a process, we will think, also about the GMP requirement. It's very important for this stage. Not only the manufacturing, but also the quality control. But I think just to know, we have a question that the basic requirement, average requirement on the GMP, I think this figure can answer that question better. We have, I think, not only the ICH, PICS, or FDA, or EMA, or other agencies, their principle or their consideration on the GMP quality system is similar. And during the product development, we use faces appropriate GMP control. That means not always if the product at the R&D stage, and if another product is in the GMP stage, the requirement is different. Requirement on the GMP is a little bit different. So this is a figure from the FDA's guideline, and it shows that when the product is at the early stage, the requirement for is wider, is less than the product if already in the commercial stage or BLA stage. If I will show you that for the analytical method, we have different requirements at early stage and BLA stage, and also for the for the material controls is also a little bit different. But ProBio also use these principles for all our material, all our products development. After the manufacturing, the product need to be tested and characterized to show that the product is safe and or they have the aimed quality attributes. So I just put an example for plasmid control and LVV control. For no matter for which kind of product, we have general properties like the appearance or pH and these items. And also we have impurity controls, and we also have some controls on residue or some burden in the toxin, like this safety controls. So for this method, we also, these two tables also list some test method for each kind of testing items. So if your target filing regions is in US, we need to comply with USP, or if it's target to Japan, so we need to follow the Japanese pharmacopoeia. So yeah, ProBio also offer the appropriate testing method and the appropriate acceptance criteria for different products. When we talk about the product quality, I would like to talk more about on the variance and the impurities. Actually, variance and it is not purities. It's the substance that have the desired properties. That means it still have the activity. But for impurities, there are two kinds of impurities. One is product-related impurities, and another one is process-related impurities. All this information need to be showed to agencies when you submit IND or BLA. Like for the because we talked that during the IND stage, the safety evaluation is the most important part. And also for the BLA, again, safety is again also important. So we need to show them whether there's like product-related impurities, like whether there's any fragments or aggregates. And for the process, yes, my colleagues talk about that. We have some steps may induce some impurity residues, like we use BSA, so there's some residue BSA, or we use antiform, so like antibiotics. So this residue should be controlled for sometimes as the product release spec release control, and sometimes tested as the in-process testing. Regarding the method, as just said that for the analytical for the method, we have like compendial method or the product specific method. For the product specific method at the IND stage, the qualification is sufficient, but for the BLA stage, we have to do a full validation. And the ProBio provided two strategies for the non-compendial method at the IND stage. One is if the method is very mature, so we can use our platform method. If we use platform method, PSQ is sufficient for IND application. But if the method, we have a lot of development work, and we have product specific method, we will do method qualification. I also listed the qualification parameters for PSQ and method qualification specifically here. And you can see if the platform method used, the qualification time is less than the product specific method. All this strategy is based on our multiple product experience, which can be accepted by different agencies. During the analytical method, during the quality control, we also need to set up the reference standard, and we also should set up the appropriate acceptance criteria. So yeah, except the release method, we also need to do stability studies, because we also have to show agencies stability data, and the stability program, including the long-term stability, like accelerate or stress condition, and also in-use stability to support the dosing protect, is also will be included in the filing package. So as long as we have this data, we have adequate stability data, we can say that whether this plasmid or viral vector can support the use in like CAR-T manufacturing or other mRNA or other products. The second part, let's talk more on the CMC work for BLA application. But although the first section, I said that is for IND application, but it's also applied to the BLA. No matter the IND or BLA, the controlled materials, the qualified materials is also needed, and the standard will be higher, and for the process need to be optimized or should show the robustness. We can talk later. And for the method or quality control, again, we have higher standard. So yeah, for the BLA stage, we need to think about this. I like this figure very much. It shows that during the product life cycle management, like when you have the primary understanding on the process or product, the product already reached to the phase 1, so we already have some knowledge. But that time, before you launch your before you conduct your phase 1, maybe initiate your clinical trial or get the IND approval is your main purpose. So but you have limited time, so maybe the process is just to use the platform. So that time we don't have enough time to think about whether the process is appropriate for the commercial. So after that, during the clinical stage, we need to think about that, whether we need additional process optimization to have a better commercial manufacturing process. If so, we will do process optimization during the clinical stage in parallel with the clinical studies. From RA perspective, we recommend to have process optimization before your pivotal study, because after pivotal study, that if you have major change, you will induce more regulatory activities or more compatibility studies. So after optimization, before BLA, we will do process characterization to help to gain additional knowledge and further support establishment of the design space. And also after that, we have a good, better control strategy. With this design space and control strategy, then we can have our PPQ run. And both that PC data, process characterization data and the PPQ data will support our BLA filing. And after BLA filing, it's not mean if the product gets the BLA approval. It's not mean that everything is finished. We need to continue monitoring our process, continue to monitor our product. If needed, we can also have a change after commercial. So this is called life cycle management about the product or process. This slide shows the process validation definition. I cited the FDA's definition here. It means that there are around three stages of three stage for the process validation. The first one is stage process design. That is the PD work development, your early development, or also including the process optimization and process characterization. And the second stage is the process qualification. We can call it like the process performance qualification, PPQ run. And for the PPQ, combine different agencies' requirement. We know it's need at least 3 lots of production. And after process qualification, we, as just said, we need to continue the monitoring the process. So we have continued the process verification. We need to see whether it needed to have additional change or others would not need. Regarding the process characterization, the typical goal of process characterization is to identify process parameters that impact on product quality and the yield. So I cited this figure from the ISPEE. We can see that when we do the process characterization, we can have 7 steps. And the first, we have the master plan, and then we have the risk assessment, like use the FMEA or other tools. And then we because the process characterization, there are a lot of experiments, so it's not we cannot do it like at the scale. So all the studies will do at a small scale. So before the experiment, we needed to set up the scale down model. After that, then we can start our studies. Now for the upstream or downstream, or sometimes including if there's DP product, we also do this one for the finish and fill and finish. And after that, after the process characterization, we have output, like as mentioned, that the design space and the control strategy. This is a figure to show that how to do the assessment for the before the process characterization, or before before we do the process characterization, we need to analysis whether this process parameter will impact the CQA critical quality attributes. If yes, then we need to analyze whether the risk is high or low. If the risk is high, we will define that parameter as the CPP, and if the risk is low, we will define it as like maybe KPP or others. But if the risk assessment, if the assessment the parameter will not impact the critical quality attributes, then we can define the parameter as a general process parameter. So yeah, this is the assessment tree for your understanding on the risk, understanding how to do the process characterization. For the process, it's a life cycle management. It's also applied to an analytical method. It doesn't mean that the analytical method, when you set up, after you validate, it will not change. No, after we have the validate the analytical method, we will still monitor whether there's new technology or whether they are have high frequency frequent QoS or other issues. We may need to think about whether the method optimization is needed. So it's the same, it's a life cycle management. For the R&D stage is a method development. We need, sometimes we use, as mentioned, that we will use some platform method, or sometimes develop the product specific method. And for R&D, we can do the qualification, but for the BLA, we need to do the validation as per guidance. And the ICHQ 14 a good choice. Here, yes, we provide summarized a table for the method validation items based on WHO, USP, ICH, and the CHP. I think that guidance almost cover the different regions requirements. You can see they are similar. Yeah, and this slide shows that just to give you an example on the LVB BLA stage CMC package. So from this table, you can see for LVB, for the BLA, at the BLA stage, we need to conduct the PC process characterization for the manufacturing process. And also we need to do method validation. And we also should do PPQ. But PPQ is not just like sometimes we said that PPQ, we run 3 consecutive production. It's no, for PPQ, there's a lot of work to do before PPQ. We have a lot of preparation work, including we need to check what's the equipment status, what's the method status, and the document, and so on. And then after we have everything ready, then we have run the 3 lots. After also, except the PPQ, the production, we have a lot of supporting validations, such as like we use cultural media. So we will validate, or we have some studies to see what's the shelf life of cultural media. Also, like because we use a lot of buffer for the downstream, so we also need to do the shelf life validation. And for the intermediates, we will do the holding time study and also like the leachable, extractable studies. And for the reasons or the memory, we'll see whether we need to do the life cycle. And yeah, after that, for the quality part, we will have the release data, and also we have a program to monitor the LVV stability trends. So we have the long-term stability and like accelerate or stress condition. Yeah, this is just an example for the LVV BLA stage CMC work. And yeah, my last part is comparability consideration. As you know, for product development, especially now many of us doing the innovative drugs. So like different stage, we have different goals. Like the R&D stage, we want to initiate our clinical trial as soon as possible, right? So we have very limited knowledge or limited data to help to set up our process sometimes, or analytical method, or we use some platform materials. But after we go to the phase 1 or after phase 1 with the oh yes, this product can be moved to next step, it to have chance to be launched. So that time we needed to think whether we needed to do some changes, change the process as more appropriate as the commercial process, or whether there's new technology to let us think the method maybe can be updated. So there's a lot of change during the clinical stage, and sometimes there's a lot of changes even after the commercial. Like sometimes we think the materials cost is too high. We want to use the local materials, which will make the can reduce the cost. So no matter the clinical stage or commercial stages, there are a lot of changes. Then if you have the changes, you need to think about the comparability, whether the pre the changes or post the changes, product is comparable. Only with this data, the agency will accept these changes, especially the changes affect the safety. So we need to think about what things we need, how to do the comparability studies. This is I think we analyze the different agencies guideline and also the ICH. I think the principle is the same. And for the clinical stage, we need to analyze the change based on whether it will impact the safety. And if we go to the later stage, we need to think about whether the quality, the efficacy will be impact. So comparability studies is a stepwise and phase appropriate principles at a different stage. Like if the product is at the early stage, we have a lot of clinical trials will be conducted in the future. Then we can have limited studies. Like sometimes we just need to do the studies on CMC part, because we will use the poster materials in the clinical trials. But the product, if already commercial, there will be no other critical clinical trials. So then if there's a major change, we need to think about, except the CMC comparability studies, we may need to do non-clinical comparability, or even sometimes the clinical comparability. And so yeah, so when you talk about the change. Sometimes sometimes I'm asked that whether we can have the change. If you have this question, the answer always yes, but we need to think about how to do the comparability studies and whether it's appropriate time to have this change, because different stage or different changes will have different comparability programs. This is analysis tree, maybe a good tree for your understanding on the change assessment. If you plan to have a change, you can check whether it will impact the quality attributes. If yes, then we need to see whether it's at the early stage or whether it's already at the phase 2B or phase 3. Then we need to think whether then to decide whether the CMC work is enough or we need to induce the non-clinical studies or clinical bridging study. But if the analysis that this change will not impact the quality attributes, then we can have a very limited analysis, a limited study. Sometimes even the paperwork is also okay, like for example, at IND stage, we want to change a vendor over raw material, and the raw material spec is the same, then maybe we can just do a paperwork to demonstrate that the material spec is the same or the higher, then we can approve that change. Yeah. So for different for this kind of change, different kinds of changes, different agencies have different filing pathway. And the first, the up table shows that the filing pathway for change during the clinical stage. And the bottom table shows that the filing pathway for the post-approval change. I think the principle is similar. And for the clinical stage, thereon can be divided into two parts. We need to think whether it will impact the safety. Both FDA or EMA or even NMPA, I believe other agencies also have similar consideration. If the changes may have impacted the safety, we need to report to agency and get their approval. But sometimes it mean not mean like FDA and not mean that get their approval, but we will wait for 30 days. If there's not any objective from FDA, then we will exclude the change. But if we analysis and we conclude that the change will not impact the safety, we can just report to agencies as an annual report. That means we can do the changes immediately. No need to wait for the agency's feedback. For the post-approval changes, there are most agencies, they will have 3 categories. The first one is the major change, and the second is the moderate change, and the minor change. For the minor change, the same as the clinical stage, if the changes have no impact in the safety, we can just report to agencies as annual report. But if the change is major or moderate, we have different filing pathway, but they are similar that we'd better to wait agencies feedback. And for the major changes, we need to get the agency's approval before implement that changes. That is all the changes we need to think about. from the regulatory perspective. Yeah, that's all the information I'd like to share with you today. Thank you.
Thanks for Tina's great sharing. Let's move to the Q&A part. And I found there are two more questions about our second topic. So I want to invite Dr. Lu to answer these questions again.
Yeah, no problem.
Yeah.
Okay, what's the question?
Yes, it's in our chat box. The
Okay,
first question is it acceptable from a regulatory perspective to switch from adherent LV to suspension LV in phase 2?
Yeah, for this to be acceptable, apparently there's gonna be have to be comparability studies for the quality and stability of the lentiviral vectors manufactured, but then also comparability study at the T cell level on the client side. So if the data of the comparability study show that the LVV before and after the process change are comparable, then this change in process can be considered acceptable by the regulatory entities. But if the comparability studies at the T cell level cannot prove their comparability, then additional non-clinical animal study or clinical bridge studies will need to be required to convince the regulatory entities to accept this change.
Thank you. Does Tina has any more comments on this question, since it's also related about regulatory?
Thank you. Yes, I totally agree with Dr. Lu. Actually, ProBio did such a case, and that project already under the agency's review. And as I mentioned, that we need to evaluate whether this change will impact the safety or the efficacy. For such a kind of change, we defined it as a major change. This may impact the safety. So for such kind of change, we will recommend to have an filing activities. That means we need to submit the comparability data to agency to get their approval.
Thank you for your two answer. And next question still belongs to Dr. Lu. In LVV manufacturing, the final fill volume will be less, but has this led to an increase in manual operations? Additionally, how has the risk assessment regarding contamination management from these manual operations been evaluated in terms of GMP?
So manual operation compared to like an automated filling, operation can sometimes help reduce material loss during the aseptic filling process. It is acceptable if the LVV is, used as a material in creating cell therapy, especially when the batch size is relatively small. So, and also many of the commercially available products do use manual filling for their LVV. The manual filling operation can be performed in isolators along with using single-use flow path to minimize the risk of contamination or cross-contamination. But overall, we still think that automated filling systems is the preferred method of filling.
Thank you. Thanks for Dr. Lu's answer again. And next question is belong to Tina. What kind of information about materials need to be provided to agency when submit IND?
Yeah, thank you for the question. And for the materials, as I mentioned, that especially there may be different kinds of materials used. Like for the excipients, we need to have the for the we draft the IND package or BL package based on the CTD format. In that CTD format, we have a separate section for the excipients. So we need to provide the specification, like the method method validation, release data, and also the justification of the specification. This information of excipients need to be provided. And like for the container closure system, also we have a separator section and the spec of the container, the vendor information. This information need to be provided. But for other materials, also we need to think to whether it's a critical materials or whether it's human or animal origin materials. If so, we also need to provide like TSE, BSE statement to show that the risk of the TSE, BSE is controlled. There's no impact on the safety. So this information need to be provided. And besides, we need to provide like the COA of the materials and to show they are well controlled. Yeah, I think this information.
Thank you. And the next question, I think it's similar as previous one. Do we need to perform clinical studies when change manufacturing site of products?
Yeah, it's also a question related to the changes. What kind of studies need to be performed when we have the change? Here, it's the changes about the manufacturing site. I would like to say the answer is depend. If the manufacturing site change at a very early stage, and like if your change is a plasmid sometimes, then I would like to say there's no need to conduct clinical trials for such kind of change, because as long as you, of course, your CMC compatibility data show that the comparability data is very good, is good. And but if the change, the manufacturing site change happened after your pivotal study, that means you have no more clinical trials to demonstrate that the post change materials in human. So then maybe a clinical bridging studies will be required by agency. So yeah, so it depends on which stage of the clinical site change. So from regulatory perspective, we will recommend that if you have manufacturing site change plan, better to do before your pivotal study.
Thanks for your sharing. And the next question is, for an ex vivo BLA application, how does ProBio establish the acceptance of COAs, such as titers, HCP, and HCD, or do they simply report the results?
That's a good question. Actually, I was asked by some persons about that, and now I think we have a very clear answer that for even the HCP, HCD or titer, if that one is in your release control panel, we should have the limit. That means we should have a actual limit for these items. But if there are some items not as your release panel, then you can use the report result. But if that is the release panel, you have to set up the limit. This requirement, I think, many times ago, many years ago, we have a lot of projects that just report result to and then to file to FDA. FDA have rounds of feedback that please set up the criteria, the limit for the release test item. If you just at the very early stage, the range can be wider, but you have to have the limit, because you want to control. If there's just a report result, we cannot control.
Thanks for Tina's answer. That's all the questions I received by now. If you have any further questions, feel free to email us later.